In this video, I'm going to talk about different techniques in molecular biology. So this is the very common topic which is asked in all the CSR exam, DPT, ICMR, and even for interviews, the questions come from these different techniques, how you identify the interaction between proteins, how you identify different proteins and DNA and RNA. So before going ahead, please like, subscribe and share this video. So in this video, I'm going to cover these topics: agarose-gel electrophoresis, southern blotting, northern blotting, western blotting, and electrophoretic mobility shift assay. So I will go in detail about each of the technique, and if you have any question, please let me know in the comment section. So first technique, which is an agarose-gel electrophoresis. So this technique is used to separate DNA based on their molecular weight. So I think all of you know that DNA are negatively charged, and when you load your samples, which you can isolate DNA from cells, from bacteria, and then you can amplify them using polymerase chain reaction. And once you have enough DNA, you can load this DNA in this gel, which is made up of agarose. And because the DNA is negatively charged, you apply current and then the DNA will move from negative charge to positive charge. So because all the DNA is negative, the only difference between this DNA is the size. So based on this size, DNA will move at different rates. Like the lower molecular weight, lower size will move faster as compared to the larger size. It's because agarose gel, when you use the agarose gel to separate your DNA sample, it will form pores upon solidification. And the smaller the pore, the only the smaller size molecule can pass through it. But for the larger molecule, it will cause resistance and the larger molecule cannot fit in this agarose gel. So based on this, you can separate molecules based on their size. So the larger molecule will stay on top and the smaller one will move at the bottom. So this technique will help you to separate DNA based on their molecular weight. But with this technique, you cannot identify what specific DNA is there, like the sequence of DNA, or this is the DNA which is of your interest. You cannot identify this. So to identify this, you have to move to another technique which is called southern blotting. So this technique is in the beginning is the same, where you have to separate DNA by size using the gel electrophoresis, which I have already explained. But then to identify that specific DNA, you need the probes which can bind to that DNA only. So I will explain you. So first step, you do the same. You separate the DNA by size via the gel electrophoresis. And once the DNA is run on the electrophoresis, you transfer them to the nitrocellulose membrane. So these membranes, they are mainly the charged membranes, and upon putting them on the agarose gel and applying current, and based on the gravity and everything, this DNA size molecule will move to the nitrocellulose membrane. So you can see, you apply the membrane to the agarose gel and then you apply the current, and this DNA will move to the membrane. And once you have the membrane with this bound unlabeled DNA, because this is not labeled, you put the sequence of the specific DNA that you want to see. Suppose you have a sequence of ATGC, and then you can put a probe which is complementary to this, which is, as you know, the complementary to A is T, to T is A. So based on this complementary relationship, you can put the probes which will bind specific to this DNA. And these probes are labeled with radio, it is radio labeled. And then you can identify and check this radio labeled probes by autoradiography. And based on that, you can identify specific DNA of your interest. So this technique will help you to check if the DNA you are interested in is present in your sample or not. Suppose you have some DNA of viruses that you want to check whether this sequence is also present in humans or not. So based on this technique, you can identify that. So now we have talked about DNA. But there is one more nucleic acid which is RNA. So there should be another technique to identify the same. We are identifying for DNA for RNA as well. And this technique is called the northern blotting. So this technique is used to detect specific RNA molecule. It's the same as DNA, but the only difference is you separate RNA. So, one thing, DNA is very stable and it will not degrade very fast like RNA. So, for RNA, you have to be very careful when isolating, and it's also very prone to degradation and contamination. So whenever you are working with RNA, make sure that you are clean your bench area very well with RNase free water and everything, and make sure that there is no RNase around it. Otherwise, you will not see any band in this agarose gel. So be careful when isolating RNA. Always use fresh gloves, clean your bench area, and always try to use the RNase free water. So for this you separate RNA by gel electrophoresis like we did for the DNA, and then the procedure is exactly the same. You transfer them to nitrocellulose membrane because RNA is also negatively charged, and then you based on the current and gravity you transfer them to the nitrocellulose membrane, and once it is bound you put the DNA sequence which is complementary to RNA, which is also probed with the radioactivity, and then you can identify them using autoradiography. So the technique is same. It's just that you have to isolate. There are different protocols to isolate DNA and RNA based on their pH, based on their like they form different pH because one contain one OH group, another one contain one less, and everything. And now, so we are done with the nucleic acid, and now we move to the protein. So with protein, it's very common to use Western blotting, and this technique is very helpful to detect the protein of your interest by using antibodies. So like we were using the radio labeled probes, in this case you can use the chemiluminescence approach and then you can use antibody. So first thing is you isolate your protein by different methods, which can be repa-lysis buffer, which can be SDS buffer, which can be NP40, HIPAS buffer. So there are these different buffers available through which you can isolate protein based on the what you want to isolate, like you want to isolate nuclear fraction, you want to isolate cytoplasmic fraction, you want to isolate specific mitochondrial protein or the ER protein. So based on your requirement, you have different protocols available that you can use. And once you have the DNA, once you have the protein, what you add is SDS. Because if you know the protein is not always negatively charged like DNA and RNA. Like in case of DNA and RNA, they are negatively charged, so the only difference between them is the size. But in proteins, proteins can be positively charged, negatively charged, or neutral. And then the another difference is the size. So this is too complex and it can be like 2D in which once you are checking the charge and then you are checking the molecular weight. So just to simplify this, what we use is STS. So STS is called sodium dodecyl sulfate. It is negatively charged. And when you add this SDS to your protein sample, all the protein will become negatively charged. So because all the protein is negatively charged, now the only difference between them is the size, the molecular weight. So once you have this negatively charged protein by SDS, you can separate them on two different gels. One is called the denatured gel, another one is called the native gel. So if you know, the denatured gel is also called the SDS page gel because in this you make all the protein negative and it's all linear, because if you change the charge on the protein become disfolded and it become linear, and then it's easy to separate them. And in the native page you do not add any charge and the protein will run on the native page based on size and charge everything. So once we have the negatively charged protein based on SDS, or you have the native protein which you run on native gel. So you can run this protein, and how this protein will run in the SDS space. There are basically two type of gels in between this. So the first one is called the stacking gel, in which the all the protein will come at a particular layer, because the SDS is almost the same as the agarose gel, like it's also acrylamide, polyacrylamide gel, and it also forms spores. So the more the polyacrylamide you use, you will have smaller pores. So imagine if you have tons of people in one room, then there is not much space for other people to pass. That's the case. So if you have high percentage of polyacrylamide, there is very small pores available through which only smaller molecular weight of protein can pass. So this is the same concept for STS gel. First you have the stacking gel, and in this stacking gel there is nothing resolved. So the stacking gel is just to bring all the protein to one layer, so that after this point, they will only move based on their molecular weight. So as you can see, these wells, just below these wells, the wells included is the stacking gel, and after this is the resolving gel. And in this resolving gel, the proteins will move only based on their molecular weight. So higher molecular weight, because of the percentage of the gel, and because they are larger in the weight, they cannot cross the pore very easily, and the smaller molecular weight can go and pass very easily, as compared to the larger one. So once you run this protein on this gel, exactly the same way like we did for southern and northern, you can transfer them to PVDF membrane or nitrocellulose membrane based on your protein. And once you transfer them, we can put antibody, which is like primary antibody, which can recognize the proteins in your membrane. And there are could be like only primary antibody, which is tagged to the fluorescent protein, and then you don't need the secondary antibody. But the most common approach that is used in most of the labs is you put primary antibody, which recognize the protein of your interest on the membrane, and then you incubate again with the secondary antibody, which will bind to the this region. You see the constant region of the primary antibody, and then you add another substrate, because this secondary antibody is tagged with one of the protein which is called HRP. And once you add this protein substrate, so this HRP is an enzyme. So once you add this substrate, wherever the enzyme is present, because this enzyme will be present only if there is primary antibody bind. So wherever this enzyme is present, the substrate will go, and the product is released, which forms a band. So then you can add these and check them in the chemiluminescence imaging system, and then you can see the band at specific places of your interest only. And now we have talked about protein detection, DNA detection, RNA detection. But what about proteins that interact with DNA? Or how do you identify the protein and DNA interaction? So there is another technique which is basically used to identify the DNA and protein interaction, which is called electrophoretic mobility shift assay. As the name suggests, it has to be related to something, the shift in the movement of the protein or shift in the movement of the bands. So what we do is we take the protein sample, we take the DNA sample, and then we mix both of them. And if there is any interaction between protein and DNA, they will form interaction, and when you run them on SDS gel, because of the interaction, the molecular weight, the size of the protein and the DNA will increase, because suppose the DNA is of 100 kilodalton and the protein is also 100 kilodalton. If they merge, it will be 200. But if they are not interacting, they will be individually 100 and 100. Because of the increase in the size, because of their interaction, they will move slower, like I have explained in the Western blotting and in the DNA in the Southern blotting, that higher the molecular weight, they will be on the top because they will not move much faster. So if they are interacting, they will be of high molecular weight and will stay at the top. And then you can see the shift in the probe. So here, as you can see, this is the protein and then this is the inter-DNA. And if they are interacting with each other, there is shift in the band, right? You can see the free probe, the shift and super shift, because if they are interacting, it will be super shift, and then you can identify if they are interacting or not. Okay, thank you. Please like, share and subscribe my channel.